Number 1. The Siberian crater plague factories. In 2014, satellite analysts scanning the Yamal Peninsula in northwestern Siberia noticed something that shouldn't have been there. A hole. 30 meters wide, 50 meters deep, blown clean through the tundra floor by a pocket of pressurized methane gas that had been building beneath the permafrost for centuries, until the frozen lid above it got too thin and the whole thing detonated. Within 2 years, dozens more appeared. Some were small enough to miss from the air, others swallowed entire hillsides. And the craters weren't just releasing methane, they were ripping open layers of frozen earth that had been sealed shut since the Pleistocene. The largest is the Batagaica crater in Yakutia. It's roughly 1 kilometer long and 86 meters deep, and it's growing by 10 to 15 meters every year. Its walls are a cross-section of time itself, 200,000 years of frozen sediment stacked in visible bands, each one containing organic material, ancient soils, and the preserved remains of Ice Age animals. Horses, bison, mammoths, all of them carrying their own microbial ecosystems, frozen mid-decay. Siberian permafrost covers approximately 23 million square kilometers and contains an estimated 1.5 trillion tons of organic carbon, twice the amount currently in the atmosphere. Some of it has been continuously frozen for over 650,000 years. Russian scientists at the Institute of Physicochemical and Biological Problems in Soil Science have documented viable bacteria in permafrost cores dated to 3.5 million years old. Not fossil traces, living cells, dormant, frozen, and waiting. The European Space Agency's Copernicus program tracked over 7,000 new thermokarst features forming across Siberia between 2010 and 2022. The Arctic is warming 2 to 4 times faster than the global average, a phenomenon called Arctic amplification. And in the Chursky region of northeastern Siberia, the seasonally thawed surface layer has deepened from 30 centimeters in the 1970s to over 90 centimeters today. Underground peat fires called zombie fires smolder through entire winters beneath the snow, accelerating localized thaw from below. In 2021, Miner and colleagues published a warning in Nature Climate Change. Thawing permafrost could release ancient bacteria, viruses, fungi, and protists that modern immune systems have never encountered. By 2022, Jean-Michel Claveyri's team at Aix-Marseille University had proved this wasn't theoretical. They pulled 13 distinct ancient viruses from 7 different Siberian locations, all still viable. Radosha's Rospotrebnadzor has identified over 13,000 historical burial sites for anthrax-infected livestock, many sitting on permafrost, that is now losing its structural integrity. A 2023 paper in PLOS Computational Biology modeled the epidemic risk of what they called time-traveling pathogens, and found that even low-probability release events could trigger outbreaks in populations with zero immune memory. The craters are just the entry point. What matters is what's inside the frozen ground they're tearing open, and some of what's been found down there has survived longer than any living thing should. Number 2. The 250-million-year-old bacteria sealed in Permian salt crystals. In October of 2000, a paper appeared in Nature that should have rewritten biology textbooks. Russell Vreeland, William Rosenzweig, and Dennis Powers announced the isolation of a viable bacterium from a fluid inclusion Sowie inside a halide crystal extracted from the Permian Salado Formation in Carlsbad, New Mexico. The crystal was 250 million years old. The bacterium was alive. The sample came from 569 meters below the surface, inside a borehole at the Waste Isolation Pilot Plant, a U.S. Department of Energy nuclear waste repository chosen specifically because the salt formation was considered biologically inert. The irony was immediate. The bacterium, designated strain 293, was closely related to Bacillus merismortui, a salt-tolerant species still found in the modern Dead Sea. 250 million years of apparent lineage continuity, a living organism sealed before the Great Dying, before the dinosaurs, before the Atlantic Ocean existed. Fluid inclusions in halite are microscopic pockets of the original brine from which the crystal precipitated. They form sealed microenvironments, no air exchange, no water infiltration, no external energy input that can preserve chemistry and apparently organisms for geological timescales. Freeland's team used 11 different sterilization steps and multiple negative controls to rule out contamination. Critics challenged the results anyway. Alan Grauer argued the DNA sequences were too similar to modern species. Others pointed to the theoretical limit of DNA survival, estimated by Allentoft and colleagues in 2012 at somewhere between 1 and 10 million years under optimal conditions. 250 million years was orders of magnitude beyond that. But the results didn't stand alone. In 2005, Schubert and colleagues found additional viable prokaryotes in Permian and Silurian salt deposits from Michigan and Kansas, the Silurian samples dating to 430 million years ago. In 2020, Yakola and colleagues revived halophilic archaea from 390-million-year-old Devonian rock salt in Kauri, Pakistan. One proposed mechanism for survival at these timescales is not continuous dormancy, but ultra-slow metabolic cycling. The organisms may use trace nutrients within the fluid inclusion to repair accumulated DNA damage over millions of years, never truly shutting down, never truly waking up. The waste isolation pilot plant was built to contain radioactive waste for 10,000 years. Freeland's bacteria had been alive for 25,000 times longer. If non-pathogenic bacillus can survive inside geological formations for 250 million years, the question isn't whether pathogens could do the same. The question is where else we haven't looked. Salt mines in Germany, Poland, and Iran contain fluid inclusions spanning 250 to 600 million years that have never been systematically screened, and those formations are being actively mined. The scale is almost incomprehensible, but you don't need to go back 250 million years to find something terrifying. 4 million years in a sealed cave is enough, and what's been found there is already resistant to our best drugs. Number 3. Lechuguilla Cave's 4-million-year-old superbugs. Lechuguilla Cave sits beneath Carlsbad Caverns National Park in New Mexico. Discovered in 1986, it has been mapped to over 240 kilometers of passages and descends 489 meters, making it the deepest limestone cave in the United States. It is also one of the most isolated ecosystems on the planet. The cave has been sealed from the surface for an estimated 4 to 7 million years. No natural opening large enough for meaningful airflow, water infiltration, or animal entry existed until human explorers broke through. In 2012, Kirandeep Bhullar, Jerry Wright, and colleagues at McMaster University published a study in PLOS1 that should have alarmed everyone who read it. They cultured bacteria from deep within Lechuguilla, Pentabacillus species living on cave wall biofilms at depths that had never been exposed to a single human-made chemical, and found that these organisms were resistant to 14 different commercially available antibiotics, including daptomycin, a synthetic lipopeptide that didn't exist until 2003. The resistance mechanisms weren't novel. They were the same ones found in hospital superbugs: enzymatic degradation, efflux pumps, target modification. The exact molecular toolkit used by MRSA and vancomycin-resistant enterococci to survive in intensive care units had been sitting on a cave wall for 4 million years. In 2016, Pawlowski and colleagues, also at McMaster, pushed further, identifying over 400 distinct antibiotic resistance genes in the cave bacteria, many of which had never been documented in any clinical setting. Some of those genes were structurally identical to resistance genes found in modern hospital pathogens, suggesting that horizontal gene transfer between environmental and pathogenic bacteria isn't just possible, it's ongoing. Hazel Barton, a microbiologist at the University of Akron who has studied Lechuguilla's microbiology since the early 2000s, coined the term resistim for the complete collection of resistance genes in an environment. Lechuguilla's resistome isn't a response to human antibiotic use. It's the default state. These bacteria survive on chemolitho-autotrophic metabolisms, deriving energy from iron, manganese, and sulfur compounds in the rock, and they produce their own novel antibiotic compounds to compete with neighboring species, and they simultaneously carry resistance to those same compounds. The arms race between microbial competitors has been running for millions of years longer than our pharmaceutical industry has existed. We didn't start this fight, we walked into the middle of it. The cave isn't unique. Similar ancient resistant bacteria have been found in Moval Cave in Romania, sealed for 5.5 million years, in Frasassi Cave in Italy, and in the Bolby Mine in the United Kingdom. sitting in 250 million-year-old Permian salt deposits. Fewer than 200 people have ever entered Lechuguilla. The vast majority of its bacterial species have never been cultured. They're known only from DNA sequences. What lives in the dark is armed, ancient, and largely unknown. And the fossil record shows what happens when biological defenses collapse on a planetary scale. 252 million years ago, fungi inherited the Earth. Number 4. The End-Permian fungal spike. The End-Permian mass extinction, approximately 252 million years ago, was the worst biological catastrophe in Earth's history. 96% of marine species and 70% of terrestrial vertebrate species were annihilated. The cause was the Siberian traps, the largest known continental flood basalt event, which erupted roughly 3 million cubic kilometers of lava over the span of about 1 million years, pumping colossal volumes of carbon dioxide, sulfur dioxide, and methane into the atmosphere. Oceans acidified, temperatures spiked, oxygen levels plummeted, forests burned, and then didn't come back. The coal gap, a period in the geological record where no coal-forming plants existed, lasted nearly 10 million years. And in the immediate aftermath, something else took over. In 1996, Henk Vischer and colleagues at Utrecht University published a paper in the Proceedings of the National Academy of Sciences analyzing the Permian-Triassic boundary in the southern Alps of Italy. They found that the palynological record, the microscopic spore and pollen assemblage preserved in the sediment, was dominated by a single organism, Reduviae sporonites. Originally classified as an alga, Vischer's team reclassified it as a fungus based on FTIR spectroscopy, confirming chitin-like compounds in its cell walls. The same spike appeared at boundary sections in Greenland, Australia, India, Pakistan, Antarctica, and South Africa. It was global. The interpretation is both simple and horrifying. After the mass extinction destroyed plant and animal life across the planet, fungi, Earth's primary decomposers, erupted in a planetary-scale bloom, feeding on the dead biomass of a collapsed biosphere. For tens of thousands to hundreds of thousands of years, the dominant visible life on land was fungal. The fossil record doesn't show a slow recovery, it shows a takeover. Now, consider the modern parallel. In October of 2022, the World Health Organization published its first ever fungal priority pathogens list, 19 species identified as critical threats to global health. Candida auris, first identified in Japan in 2009, has since spread to over 40 countries. It's resistant to multiple antifungal drugs, and unlike most fungal pathogens, it thrives at temperatures up to 42 degrees Celsius. That detail matters. Mammals have historically been protected from fungal infections by a thermal barrier. Our core body temperature of 36 to 38 degrees is too high for the vast majority of fungi. Only about 500 of an estimated 6 million fungal species can infect humans. Arturo Casadevil at Johns Hopkins argued in a landmark 2010 paper in PLOS Pathogens that the rise of mammals after the KPG extinction was partly enabled by this thermal barrier. Fungi couldn't follow us into warm blood. But warming is changing that equation. In 2023, Casadeval's group demonstrated that Cryptococcus neoformans evolved higher thermotolerance within just 250 generations when exposed to gradually increasing temperatures. The barrier is eroding. Fungal infections already kill an estimated 1.5 million people annually, more than malaria. And the antifungal drug pipeline is critically thin. Only 3 classes of systemic antifungals exist compared to dozens of antibiotic classes. A 2024 Lancet Infectious Diseases Commission warned that the combination of climate change, expanding immunosuppressed populations, and agricultural antifungal overuse is creating the conditions for what they called a fungal pandemic. The End-Permian showed what happens when fungi get their chance. The oceans hide their own version of the same threat, sealed beneath the seafloor. Number 5, the methane clathrate microbial bomb. Methane clathrates are ice-like crystalline structures formed when methane molecules become trapped within a cage of water molecules under conditions of high pressure and low temperature. They exist primarily in deep ocean sediments along continental margins and in Arctic permafrost, and the global reserves are staggering, estimated at between 1,500 and 15,000 gigatons of carbon, potentially exceeding all other fossil fuel reserves combined. The methane inside these structures was produced by methanogenic archaea, single-celled organisms from the domain Archaea that represent one of the oldest metabolic lineages on Earth. Carbon isotope signatures in the Pilbara Craton of Western Australia suggest methanogenesis has been operating for at least 3.5 billion years. These organisms didn't just produce the methane. They're still inside the deposits that trapped it. In 2006, Inagaki and colleagues published in the Proceedings of the National Academy of Sciences the discovery of viable microbial communities within methane hydrate-bearing sediments at 635 meters below the seafloor off Peru. Life persists within the hydrate stability zone itself. The Clathrate gun hypothesis, proposed by James Kennett in 2003, suggests that sudden warming can destabilize methane hydrates, triggering massive methane release that amplifies warming in a positive feedback loop. The closest historical test case is the Paleocene-Eocene thermal maximum, approximately 56 million years ago, when global temperatures rose by 5 to 8 degrees Celsius in roughly 20,000 years. Deep-sea sediment cores show a massive negative carbon isotope excursion consistent with sudden release of isotopically light carbon, exactly what you'd expect from Clathrate dissociation. It took the Earth approximately 150,000 years to recover. Under normal conditions, the biological safety valve works. Anaerobic oxidation of methane consortia, symbiotic partnerships between methanotrophic archaea and sulfate-reducing bacteria consume an estimated 90% of methane before it reaches the water column. But models suggest that 2 to 4 degrees Celsius of deep ocean warming could overwhelm this capacity. In the Laptov Sea off Arctic Siberia, Shakova and colleagues documented methane escaping from the shallow continental shelf at rates up to 500 milligrams per square meter per day, already breaching the seafloor. But the pathogen dimension is barely discussed. The microbial communities sealed within Clathrate zones include not just methanogens, but fermenters, acetogenic bacteria, sulfur-cycling organisms, and their viromes, communities of viruses that infect them. Kallmeyer and colleagues estimated in 2012 that subsurface sediments globally contain roughly 2.9 times 10 to the 29th microbial cells, a biomass comparable to all plant life on Earth. A 2021 study in Nature Communications by Dong and colleagues found novel viral families in methane seep sediments in the South China Sea that had never been classified. Rue and colleagues estimated in 2019 that only about 1% of marine viral diversity has been cataloged, and 5 countries, Russia, Japan, China, India, and the United States, have active programs to extract methane from hydrate deposits as fuel. Japan conducted the first offshore extraction test in the Nankai Trough in 2013. No environmental impact assessment for any of these programs has specifically evaluated the pathogen release pathway. The question isn't just how much greenhouse gas comes out, it's what's alive inside it. Something that's been alive for even longer was found preserved in amber, and it's spreading right now. Number 6. Tick-Borne pathogens preserved in 15 million-year-old Dominican amber. In 2014, George Poinar Jr. at Oregon State University published a paper in Historical Biology that contained a photograph most people scrolled past without understanding what they were looking at. Inside a piece of 15 to 20 million-year-old Dominican amber, fossilized resin from the extinct tree Hymenaea protera, sat a tick of the genus Amblyomma. And inside that tick, clearly visible in its body cavity, were coiled, elongated spirochete bacteria, morphologically indistinguishable from modern Borrelia burgdorferi, the organism that causes Lyme disease. Poinar went further. In 2015, he identified rickettsia-like organisms inside ticks preserved in Cretaceous amber, dating to approximately 100 million years ago, pushing the origin of tick-borne rickettsial diseases back to the age of dinosaurs. He also found Babesia-like pyroplasms, protozoan blood parasites, in Dominican amber ticks. The full tick-borne disease triad of spirochetes, rickettsia, and pyroplasms was already established by the Miocene, and the rickettsial component was running since the Cretaceous. These parasitic relationships are not recent emergences. They are among the oldest continuously operating infectious disease systems on the planet. Molecular clock analyses estimate that the genus Borrelia diverged from other spirochete lineages somewhere between 60 and 120 million years ago. The ticks themselves were already diversified. Poinar identified at least 4 genera in Dominican amber alone. The infrastructure for delivering tick-borne disease to vertebrate hosts has been evolving, refining, and adapting for over 100 million years. And right now, that infrastructure is expanding. The Centers for Disease Control and Prevention estimates approximately 476,000 Americans are diagnosed with Lyme disease annually. Ixodes scapularis, the black-legged tick and primary Lyme vector in eastern North America, has expanded its range northward by approximately 46 kilometers per decade since the 1990s. In Sweden, Lyme cases in Norrbotten County near the Arctic Circle increased fivefold between 1995 and 2020. Germany recorded over 50,000 confirmed cases in 2023. Tick-borne encephalitis is now endemic in 27 European countries. The Lone Star tick is pushing into the northeastern United States and Canada, bringing with it alpha-gal syndrome, a meat allergy triggered by a sugar molecule in tick saliva. In 2022, the first locally acquired cases of Crimean Congo hemorrhagic fever were documented in Spain and France, transmitted by Hyaloma ticks expanding into Western Europe. Climate models project that by 2080, suitable habitat for Ixodes scapularis will include most of southern Canada, with the active tick season extended by 2 to 4 weeks annually. The amber didn't reveal an ancient threat. It revealed that Lyme disease, and the system that delivers it, has been running for at least 15 million years. Every degree of warming, every expansion of tick habitat, every increase in host density amplifies a parasitic infrastructure that predates the human species by a factor of 1,000. These organisms haven't returned from extinction. They never left, and some of them literally waited in the permafrost for 46,000 years and walked away when thawed. Number 7. The Panagrolaimus Resurrection. In July of 2023, a team led by Anastasia Shatilovich published a paper in PLOS Genetics that turned a theoretical possibility into a documented fact. They had revived a nematode, a tiny roundworm, from permafrost deposits near the Kolyma River in Yakutia, northeastern Siberia. The sediment was radiocarbon dated to approximately 46,000 years before present. The organism had been frozen since the late Pleistocene, contemporary with Neanderthals. And when they thawed it, it moved. The nematode was found inside a fossilized squirrel burrow, a microhabitat that had been flash frozen and sealed intact when the permafrost encased it. The species was initially misidentified as Panagrolaimus detratophagus, a known cryptobiotic nematode. Phylogenetic analysis revealed it was something new. Panagrolaimus cholamensis, named for the river region where it was recovered. It had entered cryptobiosis, a metabolic state fundamentally different from dormancy. In dormancy, metabolic activity slows. In cryptobiosis, it ceases entirely. The organism is not slowly dying, it is biologically paused. Once thawed, the nematodes fed. They molted. They reproduced sexually, producing multiple generations of viable offspring in the laboratory. Genomic analysis showed that Panagrolaimus cholamensis possesses a unique trehalose biosynthetic pathway. Trehalose is a sugar that stabilizes cell membranes and proteins during freezing and desiccation, acting as a kind of biological antifreeze at the molecular level. The organisms also share cryptobiosis-related genes with Caenorhabditis elegans, the model nematode used in genetics labs worldwide via the DAF2 insulin signaling pathway, the same pathway linked to longevity across multiple species, including humans. The previous record for multicellular organism revival was approximately 30,000 years, set by the same Russian research group in 2018. The 2023 finding extended that by 16,000 years. In 2018, the group also revived rotifers, another class of microscopic animal from 24,000-year-old permafrost. These aren't bacteria. These aren't viruses. These are animals with nervous systems, digestive tracts, and reproductive organs walking out of the Pleistocene. What makes this more than a curiosity is the math. Soil nematodes are the most abundant multicellular animals on Earth. An estimated 57 billion nematodes exist for every human on the planet. Many species are parasites. Parasites of humans, parasites of livestock, parasites of crops. If non-pathogenic nematodes can survive 46,000 years in cryptobiosis and resume normal life upon thawing, parasitic nematodes in the same permafrost are subject to the same physics. The Kolyma region where these worms were found is experiencing accelerating permafrost thaw. Summer ground temperatures have risen by 2 to 3 degrees Celsius since the 1980s. Background radiation models predict that DNA should degrade beyond repair within roughly 6.8 million years at 0 degrees, but practical survival in the sealed, frozen, anoxic environment of deep permafrost clearly exceeds theoretical predictions. The question isn't whether ancient organisms can come back. That's been answered. The question is, what else is waking up in the 23 million square kilometers of permafrost, now warming faster than at any point in human history? And the ground that preserved those worms also contains viruses, some of them bigger than bacteria. Number 8. Pithovirus sibericum. In March of 2014, Jean-Michel Clathrate and Chantal Abergel at Aix-Marseille University published a paper in the Proceedings of the National Academy of Sciences that should have ended any remaining debate about whether ancient viruses could survive in permafrost. They had revived Pithovirus sibericum, a giant virus recovered from 30,000-year-old permafrost near the Kolyma River in the Kamchatka-Chukotka region of Siberia. It was approximately 1.5 micrometers long, larger than many bacteria, and visible under a standard light microscope. And it was still infectious. The experiment was straightforward. They thawed permafrost cores under controlled laboratory conditions, applied the extracts to cultures of Acanthamoeba castellani, a free-living amoeba used as a safe proxy host, and watched the amoebae, lice, burst open. The virus had replicated inside them, assembled new viral particles, and killed its host cells. 30,000 years in the frozen ground had not diminished its capacity to infect, replicate, and destroy. Pithovirus belongs to the nucleocytoplasmic large DNA viruses, a group that has rewritten virology since 2003 when the first giant virus, Mimivirus, was identified in a hospital cooling tower in Bradford, England. Since Pithovirus, the discoveries have accelerated. In 2015, the same team revived Mollivirus sibericum from the same permafrost layer, a phylogenetically distinct giant virus, proving that multiple ancient viral lineages coexist in the frozen ground. By 2022, Clavery's group had revived 13 distinct ancient viruses from 7 different Siberian locations, spanning 27,000 to 48,500 years old. The permafrost isn't harboring one virus, it's harboring an entire virome, a frozen catalog of infectious agents spanning tens of thousands of years. Giant viruses are extraordinary in their genetic complexity. Pandoravirus, the largest known, carries 2,556 genes, roughly 93% of which have no match in any existing database. They are genetic dark matter, and we have no idea what most of their genes do. These viruses infect amoebae, but amoebae are not confined to laboratories. They are ubiquitous in soil, freshwater, and the human respiratory tract. Acanthamoeba itself is a human pathogen causing keratitis, a severe eye infection, and granulomatous amoebic encephalitis, a nearly always fatal brain infection in immunocompromised patients. Amoebae function as biological Trojan horses. If a giant virus infects an amoeba that subsequently infects a human host, the virus gains indirect access to human tissue. In 2020, Wirth and colleagues found 33 viral populations, including 28 previously unknown, in Greenland ice cores, extending the frozen virome discovery beyond Siberia. Meanwhile, in Yakutia, the mammoth tusk trade exposes workers to thawed Pleistocene-age material on a daily basis without any biosecurity protocols. Clathrate has been blunt in his warnings. In 2015, he published a commentary in the Proceedings of the National Academy of Sciences coining the term Pandora's permafrost. The Arctic, he argued, contains the largest single reservoir of potentially viable ancient viruses on Earth. Mining, drilling, and shipping routes are opening it before science has any idea what's inside. Viruses have no metabolism. They don't accumulate metabolic damage. Their survival depends only on structural integrity, and permafrost provides the ideal preservation conditions: cold, dark, and anoxic. The theoretical upper limit of viral survival in permafrost is unknown. What is known is that in 2016, the theoretical became actual, not in a laboratory, but on an open tundra, to a nomadic reindeer herding family. Number 9. The Yamal Anthrax Resurrection. In July of 2016, a heat wave struck Russia's Yamal Peninsula. Temperatures exceeded 35 degrees Celsius, 15 to 20 degrees above the summer norm. The permafrost thawed deeper than it had in living memory. And somewhere in the tundra, a reindeer carcass that had been frozen for approximately 75 years broke the surface. The reindeer had died of anthrax. Its body still contained viable Bacillus anthracis endospores, dormant survival structures encased in a protein coat with a core of dipicolinic acid, designed by evolution to resist extreme heat, ultraviolet radiation, desiccation, and chemical disinfection. Experimental evidence shows anthrax endospores remain viable in soil for at least 200 years. Theoretical models extend that to thousands of years in permafrost. The spores that killed that reindeer 7 decades earlier were as potent as the day they formed. The outbreak killed a 12-year-old Nenets boy and hospitalized 72 other herders. Over 2,300 reindeer died. The Nenets are indigenous nomadic people whose approximately 300,000 reindeer represent the largest herds in the world and the foundation of their culture and economy. The Russian government deployed over 250 biological warfare specialists from the RKHBZ, the Radiation, Chemical, and Biological Defense Forces, to contain the outbreak. Quarantine zones exceeding 200 kilometers were established. Nomadic families were evacuated by helicopter in what became the largest emergency evacuation in the region's modern history. The Yamal Peninsula had been anthrax-free for 75 years. The Soviet Union had maintained a mandatory reindeer vaccination program, but in 2007, Russian authorities discontinued it. The disease was considered eradicated in the region. 9 years later, the permafrost proved them wrong. The deeper problem isn't one carcass, it's 13,000. Russia's Rospotrebnadzor has identified over 13,000 registered burial sites for anthrax-infected livestock across the country, and historical records indicate that massive anthrax epidemics killed over 1 million reindeer in Siberia during the 19th and early 20th centuries. The carcasses from those events are spread across millions of square kilometers of permafrost. Many burial sites are unmarked. Records were lost during the Soviet era. A 2017 study by Revich and Podolnaya in Global Health Action mapped approximately 400 of these sites as being at high risk of thawing by 2050. The Centers for Disease Control and Prevention classifies anthrax as a Category A bioterrorism agent, the highest threat tier. The United States and the Soviet Union both developed weaponized anthrax during the Cold War. What happened on the Yamal Peninsula in 2016 was, in practical terms, an uncontrolled biological dispersal event triggered by temperature. The anthrax had been there for decades. The permafrost had contained it, the climate changed, and the containment failed. In 2020, Stella and colleagues published a spatial model in geospatial health predicting anthrax reemergence risk across the entire circumpolar Arctic, identifying additional hotspots not just in Siberia, but in northern Canada and Alaska, where anthrax burial sites also exist. The 2016 Yamal outbreak was the first proven case of permafrost thaw-induced reemergence of a lethal human pathogen. It proved that everything Clathrate and others had been warning about was not theoretical. But the most dangerous thing frozen in the Arctic isn't a bacterium. It's the virus that killed more people than both world wars combined. Number 10. The 1918 influenza virus preserved in Alaskan permafrost. In November of 1918, a wave of influenza reached Breivik Mission, and in Nupiaq village on Alaska's Seward Peninsula. Within 5 days, 72 of its 80 adult residents were dead, a 90% mortality rate. The bodies were buried in a mass grave in the permafrost, and the frozen ground preserved them, and the virus inside their lungs, for nearly 80 years. In 1951, a 25-year-old Swedish-born microbiologist named Johan Hultin traveled to Breivik Mission and exhumed tissue samples from the grave. His plan was to culture the virus. He failed. The technology of the 1950s simply couldn't recover and viral RNA from frozen tissue. Hultin moved on. For 46 years, the samples sat unused. Then, In 1997, Hultin read a paper by Jeffrey Taubenberger at the Armed Forces Institute of Pathology in Washington, D.C. Taubenberger had extracted partial 1918 influenza RNA from formalin-fixed autopsy samples, the first fragments of the deadliest virus in modern history. Hultin, now 73 years old, contacted Taubenberger and offered to return to Alaska. He flew to Breivik mission alone, at his own expense, and reopened the mass grave. This time, he found what he needed, a body he named Lucy, an obese woman whose body fat had insulated her lung tissue from ice crystal damage. The preservation was extraordinary. Lucy's lung tissue yielded complete high-quality 1918 influenza RNA. Taubenberger's team used it to sequence the entire genome of the H1N1 virus responsible for the pandemic, a landmark achievement published in Science in 2005. The virus had infected an estimated 500 million people, roughly one-third of the global population at the time, and killed between 50 and 100 million. It was an H1N1 subtype of avian origin, not a reassortant of existing human and animal strains, but something entirely new that had adapted to human-to-human transmission. That same year, Terence Tumpey at the CDC used reverse genetics to reconstruct the live 1918 virus from Taubenberger's sequence, the first time in history that an extinct pathogen was deliberately resurrected. The work was conducted in a BSL-3 enhanced containment laboratory. In mice, the reconstructed virus was 100 times more lethal than contemporary influenza strains. It killed through a cytokine storm, an overreaction of the immune system that turned the body's own defenses into the weapon. This mechanism explains the pandemic's unusual mortality curve. It killed disproportionately among 20 to 40-year-olds, the age group with the strongest immune systems. The Breivik mission mass grave remains in permafrost. It is not the only one. Mass graves from the 1918 pandemic exist across Arctic Alaska, northern Canada, Greenland, and Svalbard, all in permafrost zones now subject to thaw. In 2008, researchers confirmed that additional 1918 flu RNA could be extracted from remains in Longyearbyen, Svalbard, where 7 young miners died in October 1918. Longyearbyen's local temperatures have risen by 5.6 degrees Celsius since 1971. Early 20th century coffins are beginning to emerge from the active permafrost layer. The WHO's global influenza surveillance and response system monitors circulating strains year-round, but it is designed to detect viral evolution through reassortment in animal reservoirs, not the environmental reemergence of an intact historical strain from thawing ground. Descendants of the 1918 virus are still circulating. The 2009 swine flu pandemic was caused by a reassorting containing gene segments descended from the original H1N1 strain. The genome is publicly available in GenBank, raising dual-use concerns. The sequence enables vaccine development but also provides a blueprint for reconstruction. The permafrost preserved the answer and the threat simultaneously. The same frozen ground that allowed Hultin and Taubenberger to understand the deadliest pandemic in human history is the same medium that could release it again, and the ground is thawing. What all 10 of these plagues share is a single trigger: temperature. The Siberian craters are blown open by warming. The salt crystals survived precisely because their environment was stable, but the parallel formations in permafrost zones are not. Lechuguilla's superbugs demonstrate that resistance is ancient, and the antifungal and antibiotic pipeline is crumbling. The End-Permian fungal spike happened because warming destroyed the biosphere, and fungi consumed the remains and were replicating those conditions. The methane Clathrate communities are sealed by cold and pressure, both of which are diminishing. Tick-Borne diseases are expanding because ticks follow warmth. The Panagrolaimus worms thawed and walked because the permafrost warmed. Pithovirus infected lab amoebae because a core sample from warming ground was brought inside. The Yamal anthrax killed because the summer was too hot. The 1918 flu is in the ground because the ground was frozen, and now it's not. Arctic temperatures have risen 2 to 4 times faster than the global average. The IPCC's 6th assessment report projects that 30 to 70% of near-surface permafrost could be lost by 2100 under high-emission scenarios. 23 million square kilometers of frozen ground containing 1.5 trillion tons of organic carbon and an uncounted number of dormant organisms is losing its structural integrity. And the immunological problem is absolute. Modern human and animal immune systems have no memory of pathogens that were sealed before our species existed. The surveillance infrastructure isn't ready. The WHO's global influenza surveillance system tracks reassortment, not environmental emergence. There's no dedicated permafrost pathogen monitoring program. Russia's biosurveillance is concentrated in urban centers, thin across the Arctic. Canada's Northern Contaminants Program monitors chemical contamination, not biological threats. The Yamal outbreak happened 9 years after the vaccination program was discontinued because the disease was considered gone. The metagenomic sequencing technology to screen permafrost for viable pathogens exists, but it is not deployed at scale, and screening 23 million square kilometers is financially prohibitive without targeted sentinel site strategies. Meanwhile, 5 countries are actively pursuing methane hydrate extraction. Arctic mining and shipping routes are expanding. The mammoth tusk trade in Yakutia exposes workers to Pleistocene-age material daily. Military installations, pipelines, and roads built on permafrost are buckling as the ground beneath them destabilizes. Every one of these activities is an exposure pathway, and none of them include pathogen risk assessments. The fossil record has been telling this story for hundreds of millions of years. Every mass extinction in Earth's history was followed by a biological reorganization dominated by microorganisms, bacteria, fungi, viruses, the organisms that decompose, recycle, and inherit. The End-Permian fungal spike, the KPG fern and fungal recovery, the PETM Clathrate destabilization. The pattern is consistent. When the macroscopic biosphere collapses, the microscopic one expands. Pathogens don't go extinct during mass extinctions. They go dormant. They wait in salt, in ice, in amber, in the deep biosphere. And they come back when conditions change. The current rate of biodiversity loss is comparable to mass extinction events. The current rate of warming exceeds any natural analog in the geological record. The Paleocene-Eocene thermal maximum, the closest comparison, unfolded over 20,000 years. We are compressing the same temperature shift into 2 centuries. The biological responses documented in the fossil record, fungal dominance, pathogen expansion, ecosystem collapse, operate on their own timescales. And those timescales are now being forced into alignment with ours. These plagues didn't go extinct, they went dormant. The endospores sealed in permafrost burial grounds. The spirochetes coiled inside amber ticks. The giant viruses entombed in frozen sediment. The nematodes curled in cryptobiosis inside a squirrel burrow for 46,000 years. The bacteria locked in salt crystals for 250 million years. The fungal spores that blanketed a dead planet and waited for the next opportunity. They're all still here. And the alarm clock is temperature. The planet that froze them is now thawing them. The timeline is no longer geological, it's ours.